genome evolution due to CpG mutation rate. In mammals, 70% to 80% of CpG cytosines are methylated. In mammalian genomes, CpG islands are typically between 300 base pair up to 3000 base pair. About 70% of human promoters have a high CpG content. This is a characteristic of the genes of the eukaryotes, that in most of the genes of eukaryotes there are promoter sequences, and in the promoter sequences there is 70% of the promoter sequences are that have CpG islands. CpG islands typically occur at or near to the transcription start site of the genes, which are also called as promoters. Methylation of cytosine involves adding a methyl group at the carbon number 5 of cytosine. Deamination of the carbon number 4 turns a methylated cytosine into a normal thymine. So this is actually the phenomena that what are methylated cytosine in CpG islands, that there is a methylation, addition of a methyl group to the cytosine at carbon number 5, while deamination is a process in which amino group which is present at the carbon number 4 of cytosine removes and converts it into thymine. These are the diagrams of thymine and cytosine. Here we can see that this is thymine and this is cytosine. In case of cytosine, we can see that at carbon number 4 there is amino group. How cytosine is converted into 5-methyl cytosine, and 5-methyl cytosine is converted into thymine. So deamination of carbon number 4, this is carbon number 4, turns a methylated cytosine into a normal thymine. So first we see that what is methylation. This is normal cytosine, and this is 5-methyl cytosine, where we can see that at carbon number 5, this is carbon number 1, this is 2, this is 3, this is 4, this is 5. At carbon number 5, there is addition of a methyl group. So due to this methyl group, it is called as 5-methyl cytosine. So when there is deamination, a removal of amino group, that is from carbon number 4, carbon number 1, 2, 3 and 4, from here there is a deamination. Due to deamination, 5-methyl cytosine is converted into thymine. What is deamination? This is cytosine. In case of cytosine, if there is a removal, if there is deamination, it is converted into uracil. While in case of 5-methyl cytosine, due to removal of amino group, it is converted into thymine. CpG mutation rate is high, or the mutation rate in this region, that is methylated cytosine which are present in the CpG islands, that also works as a fuel for the genome evolution. Here we can see that how mismatch, but the wrong base cannot be identified. So here is C complementary to G and C complementary to G, but this C is methylated. So there is a methyl group at carbon number 5 of this cytosine. And if there is a mismatch repair, then it will work fine. But if there is deamination of this cytosine, this deamination of this cytosine will convert it into thymine. So instead of a methylated cytosine, here will be the thymine. So when the DNA will replicate from these two strands, this T thymine will produce its complementary strand, that is adenine, and this C and G, they will produce the original strands of the DNA. So due to the process of mismatch, this cytosine, here was C to G, cytosine and guanine. This cytosine and guanine was converted into A and T, adenine and thymine. So this is also one of the reasons or causes that how nucleotide substitution occur in case of CpG islands where C is methylated. So if we do the comparison between the ancestral genome and the modern genome, in case of ancestral genomes, there were CpG sites on different points or different locations on the genome, as well as there were methylated CpG sites. But through the evolutionary time scale or through the evolution, most of the CpG methylated sites were removed, but still in the modern genome, we see that CpG island still exist in the modern genome, particularly in the genome of the mammals, and they are usually present with the in the vicinity of the promoter sequences. Substitution rate in CpG islands is approximately 15 times higher than the substitution in the ordinary Cs or Gs of the genomes. If we conclude that what is CpG mutation rate, methylated cytosines causes a high mutation rate, and these methylated cytosines, they work as a fuel for the genome evolution.